like mainly work on computational projects. I have like a wet lab project and also like what we call dry lab project. So I guess my like motivation for like my computational work, which like I'm really excited about because I guess the way my mind thinks is like kind of like logical, kind of like a computer science, math, engineering type of way of thinking. So like one paper that like I was reading over the weekend, it's actually like not super recent, but like it's about the TCGA, which is the Cancer Genome Atlas. And so
Hmm.
this paper was like one of just like a perspective and like a little bit more about the atlas. But like now there's like you can basically search like they basically like sequenced all these different tumors, like the normal DNA and also the tumor DNA. And there were like multiple papers published in Cell about like all that they found, but now it's like pretty much used as kind of like the human tumor like atlas, basically like when you do a study, then if you do it in mouse, then you could like extrapolate to humans by looking within the TCGA. So I just like really thought it was super cool because like when it started coming out, like when I was an undergrad and I was interested in engineering and hearing about like how you can just like sequence someone's genome or sequence their tumor DNA and find these different biomarkers, and like it has like important implications for personalized medicine and like treatment strategies, and yeah, biomarkers for like prevention, etc. And like that like relates to my work. So I was like really excited by that, and I like always look back to those papers and reread them. And then in terms of like my wet lab, so I do like mainly computational, but I also have like a little bit of a wet lab project. And like in the past, I did like some cool experiment that I thought was really interesting where I took cells and I irradiated them. And then when they're irradiated, then the DNA is damaged. And then there's these double strand breaks. So I basically irradiated them on different slides and exposed them to like different conditions. But these are like special chamber slides where you have like this chamber over it, over this blue part, and then you put they're like little wells, and then you put like your cells in it and expose to different conditions. And then when you want to take a microscope image, you just take off the wells and then you stain them with different antibodies.